My name is Dr. Ping Furlan. I will be giving a brief biology review today to help you prepare for the Fundamentals of Engineering exam or FE exam. The updated FE exam specifics for biology requirements have removed cellular biology portion, so you wanted to focus on other parts, namely bioconversions, toxicology, and industrial hygiene. For cellular biology, you wanted to have some basic concepts. First, cells are the smallest living units or the building blocks of all life. All cells have three things in common. They have a cell membrane that separates inside of a cell from its environment. Cytoplasma, a water-based solution containing everything found in a cell, including ions, small molecules, and large molecules. And DNA, which is the cell's genetic materials. Second, there are two types of cells based on if they have a nucleus or not. They are called the eukaryotic cells if they have a nucleus containing all the genetic materials. Eukaryotic cells are found in plants, animals, and single-cell protozoa. They are called the prokaryotic cells if they don't have a nucleus. Prokaryotic cells are found in only single-cell organisms such as bacteria and algae. Eukaryotic cells are more complex, containing additional special parts called organelles. Mitochondrium, the site for energy, endoplasmic reticulum, the site for protein synthesis, Golgi, the site for packing and transporting, and the lysosome, the site for recycling for different cell functionalities. Third, the cell membrane contains of a lipid bilayer made of phospholipids. Each phospholipid molecule has a hydrophilic polar head and a hydrophobic nonpolar tail. The bilayer arrangement allows the hydrophilic heads facing outward and contacting the aqueous solutions both inside and outside the cell. The hydrophobic tails are tucked inside the cell, making a good protective barrier between the inside and outside of the cell. The movement of materials across the cell membranes or cell transport can be classified as a simple passive diffusion, facilitated diffusion, or active diffusion. Simple passive diffusion is the spontaneous movement of small and nonpolar molecules such as oxygen and carbon dioxide from the area of high concentration to the area of low concentration. Facilitated diffusion rely on protein channels that span the membrane and make hydrophilic channels across it. These channels allow small polar molecules and ions, such as water, sugar, and ions, to pass through by diffusion. Both simple passive and facilitated diffusions do not require energy. Active diffusion requires energy and involves either a pump or a vehicle. Example includes transporting sodium ions out of the cell and potassium ions into the cell against their concentration gradient. The bacterial growth curve represents the number of live cells in a bacterial population over a period of time. There are four distinct phases of the growth curve. First, the lag or initial phase, where the bacteria are adjusting to their environments. and are not dividing. Second, the log phase where the material, well adjusted to the environment, are dividing, doubling their population exponentially. Third, the nutrient limits and waste product accumulation caused the cells to die. When the number of dividing cells equals to the number of dying cells, the growth enters the stationary phase. As nutrient level continues to decrease and the waste product accumulation continues to increase, the death rate becomes greater than the dividing rate. The population growth experiences a sharp decline and enters the death phase. This is the fourth stage. Both the first-order kinetics and the logistic equation are used to model the material growth curve. You wanted to familiarize yourself with the equations and the meanings of the symbols in the equations so you can perform calculations shown on this slide. You wanted to know that most organisms have functional parts with several levels, from low to high. This includes cells, tissues, organs, organ systems, whole organisms, etc. Bioconversions are becoming more and more important. They refer to the systems that use microorganisms to achieve desired results. This includes treating wastewater by breaking down the organic matter into non or less toxic substances, or creating biofuels or alternative fuels such as hydrogen, methane, and ethanol. All of these processes are oxidation-reduction reactions involving electron transferring. They all form more material, water, carbon dioxide, and energy. The energy produced is used to support all the functions and the reproduction of the material. When organic matter decomposes in the presence of oxygen, the process is called aerobic decomposition. The oxygen serves as the electron receptor. In the broken down products, the carbon, nitrogen, phosphorus, etc. elements are in their oxidized forms. Carbon dioxide, nitrate, phosphate, etc. When the oxygen is absent, the aerobic material uses nitrate ions as the electron acceptor to allow the breaking down reaction to take place. When organic matter decomposes without the presence of an electron acceptor, usually oxygen, the process is called anaerobic decomposition or fermentation. This is an important process that produces red wines and biofuels such as hydrogen, methane, ammonia, and ethanol. The latter allows it to be known as the renewable energy technology. Other important reactions involving organisms include the aerobic cell production using glucose and ammonia, and the familiar photosynthesis where green plants use sunlight to make sugar and oxygen from carbon dioxide and water. Most questions in this topic are stoichiometry in nature. You wanted to relate the amounts through moles based on the balanced equations. Usually the equations are given to you, or you can figure it out using the information given to you. An example is the anaerobic biodegradation of organic matter to form methane and ammonia. They give you the formulas to figure out the coefficients for the balanced equation based on the composition of the organic waste. Biochemical oxygen demand, or BOD, is used to assess water quality. It deals with the oxygen consumption in milligrams per liter by the aerobic material to break down the organic matter. For instance, highly polluted sewage with a high BOD can reduce oxygen in water, causing the death of microorganisms. The BOD decrease follows the first-order kinetics as seen here, how it drops exponentially. The BOD removal, Y sub t, equals the initial BOD, or the total organic matter, minus the BOD at time t, or remaining organic matter at time t. It is also equal to the measured initial dissolved oxygen, DO sub 0, minus the dissolved oxygen, DO, at time t. Usually that time t is 5 days. Please give a try to solve the problems here using these equations. Toxicology concerns the adverse effects of chemicals on living organisms. There are several paths a person can get in contact with a hazardous chemical, as shown here, through thermal absorption, inhalation, ingestion, and the eye. Our bodies have natural building mechanisms to eliminate toxins involving kidneys, livers, etc. When the toxin accumulation rate is less than the elimination rate, no effects will be observed. And when the toxin accumulation rate is greater than the elimination rate, adverse effects will occur. The S-shaped dose-response curve is the central concept in toxicology. It is the plot of the logarithm of the chemical dose in the unit of milligrams of the chemical get into the body per kilogram of the body weight per day against its effect. Increasing chemical dose produces increased response, but within a relatively narrow range of the dose, as shown by the sharp slope. There are a few terms you wanted to know. First, NOEL, the max dose level that produced no observed effect. 2. NOAEL, the max dose level that produced no observed adverse effect. 3. LD50 or LC50, the lethal dose or concentration at which 50% of the test animals died. From the three curves shown here, which chemical, A, B, or C, is most toxic? Hopefully you picked A. Curve A represents a cancer-causing agent with a NOEL of 0, meaning any amount intake will produce an effect. The definitions of some important terms are listed here. They are shown in your FE exam handbook. First, the reference dose RFD is defined as the no observed adverse level divided by an uncertainty factor. The uncertainty factor UF is greater than 1000 to account for the variations in sensitivity between testing animals and humans and within the human population. 2. Safe human dose is the reference dose times the mass of standard test human. 3. When potentially exposed to multiple toxins, chemical vapors for instance, you evaluate the hazard ratio for each exposure, which is the exposed dose over the reference dose, and then add all the ratios together. The total ratio must be less than 1 to be acceptable. For carcinogens, we use the lifetime probability of risk, R, to measure the risk that cancer will develop due to the exposure during a person's lifetime. It is given by the cancer slope factor from the slope of the dose-response curve times chronicle daily intake. One of the chronicle daily intakes, CDI, based on ingestion is shown here. Essentially, it's the total amount ingested over the polluted period divided by the total body weight and the total lifespan of the individual. You may see questions involving air pollution. Here, you will need to use PV equals nRT and all the related gas laws. Knowing 1 ppm by volume is 1 milliliter per meter cube is important as shown in this example. What would you do when you see a problem like the one shown here? Industrial hygiene is about protecting the health and the safety of people in the workplace. The workplace hazards may be chemical, physical, ergonomic, or biological. Effective strategies include hazard communication, such as using MSDS, Material Safety Data Sheet, container labeling, worker training, and hazard controls such as using ventilation, personal protective equipment, and limiting exposure. Again, you wanted to familiarize yourself with these terms and equations. TLV, the threshold limit value that causes no adverse effect. PL, the permissible exposure limit. If you are exposed to several chemicals over a certain period of time, then the exposure is TWA, or the time-weighted average. To find the total hazard ratio, you find the ratio for each, which is the exposed level over the permissible level, and then add them all together. The sum must be less than 1 again to be acceptable. Here are some examples for finding the average exposures with answers. See if you could do them. Finally, the sum pollution calculation. All these formulas are in your handbook. You just need to know where to find them. The sum power level L in decibel is defined as 10 times logarithm of the ratio of the sum power in watts over the reference power 10 to the negative 12 watts, or 10 times the logarithm of the ratio of the sum pressure squared over the reference pressure 20 micropascals squared. The noise dose is the ratio of the time exposed to the permitted time at that level, and you want that to be less than 100%. Please do give it a try to see if you can answer the sample questions here. With that, I would like to conclude my presentation and will be happy to answer your questions or do some problems together with you during the scheduled help session. Thank you.